Before we begin, let us prepare ourselves for the opening prayer and the national anthem.
Lord, thank you for this day, for the presence of everyone in this call. For once again, we are gathered and given the opportunity to appreciate life and your words. Forgive us for our shortcomings and cleanse our hearts as we learn and live your words and example. Father, bless our endeavors, especially today's meeting. May you guide us in our discussions, and may you enlighten our minds in every decision that we make. Give us your grace that we may effectively do our parts for your greater glory. All this we ask in Jesus' name. Amen.
Bayang magiliw, pelas ng silanganan, alam ng puso, sa dibdib mo'y buhay. Lupang hinirang, tuyat ka ng matitig, sa manlulupig, di ka pasisingin, sa dagat at bundok, sa simoy at sa langit mong bakaw, may nilagang tulad, awit sa paglayang minamahal, ang kisag ng bawat mo'y tagumpay na naninilig, ang bituin at araw niya kailan pa may di magdidilig. Lupa ng araw ng walang ipagsinta, buhay ay langit sa piling mo. Aming ligaya na pag may mga api, mamamatay ng dahil sa'yo.
A pleasant morning to everyone, especially to our speaker and audience. We welcome you to the 9th episode of the Biological Research and Services Laboratories webinar series with the theme Bio at Work, the role of plants and microbes in environmental remediation. I am Meredith Montalvo, your MC for today. To formally open today's exploration, may I call on the Director of the Natural Sciences Research Institute, Dr. Elmer S. Estacio.
Good morning. Thank you. Thank you very much, Ms. Meredith Montalvo, for that kind introduction. Firstly, I would like to thank the organizers of this webinar, the Biological Research and Services Laboratory, BRSL, headed by Dr. Pirangeli Vital and the BRSL staff, for inviting me to deliver today's opening remarks. Maraming salamat sa panyaya. As always, suking-suki. I welcome everyone to the BRSL webinar series 2021, episode number 9, entitled Bio at Work, the role of plants and microbes in environmental remediation. Today's webinar theme is on novel techniques in reversing environment damage via microbial intervention and phytomining. Don't get me wrong, I'm not an expert in this area of study. The only reason I know of this terminology is because I have been given a copy in advance of today's webinar program. Hanggang dun lang alam ko. I'm sure that like me, you will also be learning a lot of new things from a video presentation by the BRSL interns on the role of microbes in environment recovery. And more importantly, from the lecture that will be delivered by today's guest, who is definitely an expert in this field of study, Dr. Rene Claveria, from the Department of Environmental Science at the Ateneo de Manila University, and his lecture will be on phytomining via hyperaccumulator ferns. The NSRI has been preparing for an eventual reopening of its laboratory testing and training services. In the meantime, however, we have no choice but to carry on the NSR mandate of disseminating scientific knowledge using alternative means, such as this type of webinars. With this, I would like to congratulate BRSL, its head and the staff for soldiering on each time and successfully organizing each episode of this well-subscribed webinar series. Mabuhay po kayo. So with that, let us all stay safe and healthy. Magandang umaga po sa inyong lahat. Thank you.
Thank you very much, Dr. Estacio, for welcoming the audience. We all know that increased human activities, unsafe agricultural practices, and rapid industrialization all contribute to environmental pollution. This issue has been on the rise in the past few decades, posing threats not only to the environment but also to our health. With this, researchers have developed different techniques to effectively restore polluted environments in an eco-friendly approach, one of which is through microbial bioremediation. To know more about how microbes are used in environmental remediation, let us watch a video prepared by the BRSL interns.
Heavy metals, nuclear wastes, pesticides, greenhouse gases, plastics, and hydrocarbons are just a few of the many pollutants that pose environmental damage and public health concerns.
With the emerging environmental issues, there was a need to design new techniques that are efficient, low cost, and eco-friendly. How? Through remediation.
Environmental remediation is the act of removing contaminants from the soil, groundwater, surface water, sediments, and other natural materials. It makes use of biological mechanisms to degrade or detoxify the concentration of pollutants present in the environment. Two of the most common types of remediation are microbial bioremediation and phytoremediation.
Microbial bioremediation relies on microorganisms and their derivatives such as enzymes or spent biomass to reduce the environmental pollutants. These microorganisms are widely distributed in the biosphere because they have an impressive metabolic ability and can easily grow in a wide range of environmental conditions.
These microorganisms, or known as bioremediators, have enzymes that possess a remarkably wide degradation capacity due to their non-specific and specific substrate affinity. These mediate all metabolic reactions and attack the pollutants and convert them into harmless products.
Microorganisms have a seriously important job as they act as nature's garbage disposal. An example is the mechanism of the hydrocarbon-eating bacteria, Alcanivorax borcomensis, which feast on spilled oil. This species naturally propagates and becomes predominant in crude oil containing seawater when nitrogen and phosphorus nutrients are supplemented. Additionally, the dispersants used to break up the oil can help stimulate growth of the oil-eating bacteria because they find the dispersants to be tasty too.
Bioremediation can also be used in other water systems like rivers, streams, and estuaries. Lawn, fertilizers, pesticides, and other contaminants find their way into these waters as they get picked up by rainwater that runs down the land and into the water systems. Just like with oil spills, biological degraders can be used to break down these harmful substances as well.
Let's move away from the oceans and take a look at how bioremediation can be used on land. Many farms use herbicides and pesticides on their crops, but these don't necessarily belong in the soil, and they can also infiltrate both ground and surface water.
Bioremediation can be used to decontaminate soil by mixing it with compost, which is decomposed organic matter full of all sorts of, you guessed it, decomposers. Compost bioremediation can not only rid the soil of contaminants, but also produces a really healthy soil in its place.
Now that we know the applications of microbial bioremediation, it's time to know its advantages from other cleanup techniques.
Bioremediation is a natural process. Since it solely relies on natural processes, it minimizes the damage to the environment. Microbes degrade the contaminants present and in return release harmless byproducts such as carbon dioxide, water, and cell biomass.
Bioremediation follows on-site treatments. It can be carried out on the site of the contamination, reducing the potential risks to the environment and human health that can arise during transportation.
Lastly, bioremediation is a low-cost process, since it makes use of biological specimens in order to restore polluted environments. It is less expensive as compared to other cleanup methods.
However, just like any cleanup techniques, bioremediation also has limitations.
Biological processes are highly specific. There are a lot of factors that need to be considered in order to carry out the whole bioremediation process. These include the presence of metabolically capable microbial populations, suitable environmental growth conditions, and appropriate levels of nutrients and contaminants.
Bioremediation requires technological advancements. The process needs a lot of research work to develop and engineer bioremediation technologies suitable for sites with complex mixtures of contaminants which are not evenly distributed in the environment.
Lastly, bioremediation is a long process. It takes a much longer time as compared to other treatment techniques, such as the excavation and removal of pollutants directly from sites.
Microbial bioremediation is a promising approach we could rely on. With the threats of pollution to our health and the environment, bioremediation is an ounce of hope that could minimize the pollution that we produce. With more research, most sustainable approach that could provide clean water, air, and healthy soils for the future generations.
Now that we know the importance of microbes, let us now dig into other organisms that also play a role in environmental remediation, specifically the plants. And I'm sure marami ditong naging plantitos and plantitas when the pandemic started. But what's so special about these organisms and what exactly is phytoremediation? To give us a clearer and a greener understanding of this concept, may I introduce our speaker for this morning. He is an assistant professor of the Department of Environmental Science at the Ateneo de Manila University. He is a geologist by profession and has worked in different capacities in the government, mining industry, and the academe. He received his PhD degree at the University of the Philippines with economic geology as his field of specialization. His areas of expertise are in mineral exploration, mining geology, petrology, and ore microscopy. His present researches are on phytoremediation and phytomining, and on trace and rare earth elements geochemistry. Ladies and gentlemen, let us all welcome Dr. Rene Juna Arclaviria.
Uh, yeah, good morning. I would like to, thank you very much. I would like to share my slides. Okay. Can everybody see my slide?
Yes, sir.
Okay, very good. Okay, thank you very much. Thank you very much for the very nice introduction. First of all, of course, I would like to thank the Biological Research and Service Laboratory of NSRI and also to Director Estacio for this invitation. It is a privilege to be part of this webinar on environmental remediation. Okay, let's start with, let me go back to my title. Okay, what I'm going to present now is actually a part of a research project on phytotechnologies, which is funded by Bishert. This is a collaboration of various researchers like the Ateneo, of course, University of Melbourne, Xavier University, and Mindanao University of Science and Technology. The title of my presentation is Phytomining. I don't know if this is quite new to most of you. We'll try to recover copper and arsenic from hyperaccumulator ferns. Okay, the outline of the presentation provides you the topics that I will be discussing. First, we will have an overview of what phytoremediation is, with specific emphasis on hyperaccumulators. Then to the introduction and assessment of these hyperaccumulators. And we'll proceed to the recovery of copper and arsenic from these ferns. At the final point is that we try to appreciate the applications of this research. So we proceed to the propagation of these plants and use them for mine rehabilitation. So that is the main thing that we want to achieve. Okay, what is, well, we'll start with phytotechnologies. So the concept behind phytotechnology is the use of plants in providing clean, what we call clean and green solutions to pollution problems in soil, in water, surface water or groundwater. It is also the opportunity to appreciate the capability of these plants to detect, degrade, remove, and contain some of these contaminants or pollutants. Our particular interest here would be phytoremediation. There are a number of plants that normally grow or thrive in contaminated areas, and these plants are called metallophytes. These plants are capable of tolerating the contaminants in the soil by merely avoiding them or absorbing minimal amounts of these contaminants, just enough to have a normal growth and avoid phytotoxicity. There are different types of phytoremediation, and this we call them phytostabilization, rhizodegradation, phytodegradation, phytovolatilization, phytoextraction, and phytoaccumulation. The plants that specialize in the extraction and accumulation of contaminants, for example, metals, they are what we call hyperaccumulators. They're able to transport metals from the roots, which we call the below ground components, to the shoots and leaves, which we call the above ground components of the plant. Hyperaccumulators, these are plants that can accumulate metals in their above ground structures at values commonly above 1,000 ppm and may even reach to values of about 10,000 ppm. And these plants are not affected by such accumulation. These values are beyond the normal upper limits of plants in tolerating accumulated metals. For example, the metal copper. A hyperaccumulator can absorb 1,000 ppm or more of the copper as compared to a normal plant that can only absorb, let's say, about 30 ppm. Otherwise, beyond that, the plant will experience phytotoxicity. Okay, a very good example is this plant, Sibertia acuminata of the Sapotaceae family, which is identified or classified as a nickel hyperaccumulator. Okay, the latex samples that were taken from the plant were analyzed for nickel. So based on color alone, you can already see or indicate that it has very high concentrations of nickel. From the results of the analysis, the average concentration of nickel is about 6.34%. In nickeliferous laterite mining, the cutoff grade of nickel is 2%, above which is considered nickel ore, and below 2% will be considered as waste. It is apparent that we get more nickel from the plant than from the soil. Imagine 6.34% against the cutoff grade of 2%. Another example is the Bernia cernua of the Euphorbiaceae family. This was discovered in one of our opportunistic surveys in Zambales. During that survey, we used filter papers which are laden with dimethyl glyoxime solution. This is a solution that can detect a nickel content of the plant. We use it by pressing the leaves of the plant, and if the color is, let's say, about purple to reddish in color, that's what you observe from the filter. It implies that the leaves of the plant would be containing about 1,000 or even more than 1,000 ppm of nickel. So we tested all of these Bernia cernua within Zambales area, and it was consistent. So we took Bernia cernua as a nickel hyperaccumulator that thrives very well in nickeliferous laterites. We further made some studies on Bernia cernua. We did some SEM-EDX and microscopic analysis of the leaves, and apparently have shown that the nickel is stored. Okay, they are mainly stored at the epidermis of the plant. So in essence, the plant can tolerate the accumulation of nickel, but it deposits the nickel in areas which most likely would reduce the stress during the normal growth of the plant. So those are the plant capabilities in, let's say, disposing or not disposing of accumulating some of these metals. The general objective of the research is to determine the usefulness of these hyperaccumulators. We know that they are very effective in decontaminating the soil of various metals. However, there has been a constant issue on what to do with the plants as they contain relatively high amounts of metals. These metals or these plants thrive very well and will always tend to survive in contaminated soils, as these are areas that are practically their natural habitat. Harvesting and disposing them may even be an added problem. The thought of recovering the metal from the plants would be of interest to most of us. Apparently, this has been done elsewhere. I would like to cite the work of Brooks et al. 1999, wherein it came about the concept of phytomining, where hyperaccumulators can be used to extract metals, which can extract metals from the soil. And actually, they are mainly used to extract metals that are left behind or are considered sub-economical during the mining process. So for example, in nickeliferous laterite, the conventional mining tend to leave as waste the 2%, what we call nickel content of the soil. So that's sub-economical. But if you put in the hyperaccumulators there, it can absorb more of the nickel, so it gets beyond the 2%, as I've shown you in an example earlier. So that is what we call the concept behind phytomining, using plants to mine or to extract more of the nickel. So we went to the process of assessing what we call the different types of possible accumulators that can actually be found in certain mines here in the Philippines. So we tried to assess the types or the kinds of hyperaccumulators that can absorb copper and arsenic. So we were not interested mainly on nickel. We'll try to, there are more copper and gold mines here in the Philippines. So we try to use copper and arsenic as our priority metals. So we'll use some of these hyperaccumulators in the recovery processes. So in Benguet, in a small-scale mining area at Camp 6 along Cannon Road, we noted the abundance of Pterogramma calomilanus and a few of the Pteris species, particularly the Pteris vittata. The analysis of the above ground and below ground components showed that the plants has a bioaccumulation factor of copper which is greater than 1, and a translocation factor of copper which is less than 1. And for arsenic, for the accumulation analysis, we noticed that the bioaccumulation factor for arsenic for these two plants is greater than 1, and also the translocation factor for arsenic is also greater than 1. What are these bioaccumulation and translocation factors? The bioaccumulation factor is the ratio of the plant between the plant and the soil. Okay, so numerator ay yung plant, denominator ay yung soil. So if you get greater than 1, it means that you have more metals in the plant than the soil. While a translocation factor is the ratio between the above ground component of the plant and the below ground component of the plant. So it means that if you have a ratio, translocation factor ratio of greater than 1, it means that the above ground components would likely have more metals than in the below ground components or the roots, meaning the plant can actually transport most of these metals from the roots to the shoots and leaves. We proceeded to another area, also in Benguet. This is the old mining area of Benguet Corporation. And apparently we noticed more of the Pteris vittata. So the plant, we noticed that cannot accumulate much of the copper, but it was surprisingly noticeable that the plant can absorb a lot of arsenic, particularly in the above ground components, as well as some of the below ground components. Previous studies have indicated that Pteris vittata and also the Pterogramma calomilanus, which I mentioned earlier in the previous slide, are well-known arsenic hyperaccumulators. This is well studied worldwide, and apparently our observation in Benguet Corporation confirms that indeed it is actually a nickel hyper, or what we call this an arsenic hyperaccumulator with a translocation factor of greater than 1. Sorry for. Okay, I think my slide has. Okay, sandali lang. Okay, sorry for that. Apparently, this is the only slide that got into some displacement. Sorry about this. Anyway, I think there's a small part you can still observe. So this is uh, a work we did in the south at Surigao. We went to Manila Mining, which is another copper gold gold mine in the south, and when we were doing work there, we observed a new type of fern. And this is we call the Pteris milano caulon. We haven't seen this fern in the north, in Luzon or in Benguet. Apparently, we observed most of this here at the south, sa Mindanao, particularly in Manila Mining. Although it thrives also very well with Pteris vittata and Pterogramma calomilanus. Now, we analyzed this type of fern for copper, and it showed that the bioaccumulation factor is greater than 1. And apparently, the high concentrations of copper found in the roots reached to about 15,000 ppm. It was amazing. It was something that we have not noticed in different types of plants. 15,000 ppm accumulated copper accumulation in the roots of the plant. The translocation factor of Pteris milano caulon is also, well, it's not much of the as like what it absorbs in the roots. So it is a bit less than 1, the translocation factor for copper. So since it stays only within the roots, the copper stays only in the roots, it is classified as a phytostabilizer. So it doesn't, there's no movement of the metal from roots to the shoots. Okay, then apparently we analyzed this also for arsenic. So we noticed that the translocation factor for arsenic is about greater than 1. So it means that it has the capability of transporting arsenic from the roots to the shoots, stem and leaves. So apparently, we can classify this, if there's a very good extraction capability, as a hyperaccumulator, arsenic hyperaccumulator. Okay, so the bioaccumulation factors of the plant with respect to arsenic is also greater than 1. Noting, of course, that the what we call this, the translocation factor is already greater than 1. So we thought this could be a new type of fern that can actually be used for arsenic recovery. We try to confirm all our observations. We try to confirm most of our observations using potted experiments. So actually, this is an additional thing on research to confirm analytical or chemical analysis. You confirm it with a potted experiment. So different concentrations of copper and arsenic is actually inputted into the different pots where these plants normally grow. So depending on the concentration, we would actually note that how much these plants would accumulate into its anatomical system. So using potted experiments, we confirmed that Pteris vittata and Pterogramma colaminatus are not really good accumulators for copper. They can accumulate, but not much of what we require them, more likely for extraction purposes. However, we got very much interested on Pteris melanocaulon because we noticed that aside from being a phytostabilizer, it can accumulate so much copper in the soil. It can also accumulate so much nickel in the above ground components. So it was quite amazing because we found a fern that can both accumulate copper and arsenic. Actually, this is a discovery that we had, and we published it for in a certain paper, and apparently we got some media attention on this. So some of the media has actually mentioned the discovery of this plant, and apparently was also not known worldwide that indeed we had a fern that can accumulate two types of metals, copper and arsenic. So we thought this would be a good chance for us to use this type of plant, indigenous in our area, for mine rehabilitation purposes. This slide just shows you the summary of the different analysis we made. So it shows you the different capabilities of the ferns, Pteris melanocaulon, vittata, and Pterogramma colaminatus, on how they absorb copper and arsenic. So we have chosen these three types of ferns for our recovery experiments. So we now go into the recovery process of the metals from the plants itself. Pteris melanocaulon, Pteris vittata, and Pterogramma colaminatus. The first experiment we did was to recover copper. Okay, we used the roots of the Pteris melanocaulon, as I've noted earlier. It can reach to about 15,000 ppm. So that high concentration, we need to extract that from the plant. So we try to recover it by what we call the electrowinning process. Electrowinning is a very old traditional method used in mining and metallurgy. Actually, if you go to a mining area, they are using large-scale electrowinning equipment. So we're going to use this because it is a very effective method of extracting copper. So the method is relatively simple. We use two electrodes, an anode and a cathode, and we apply current like that. So the solution that we prepared is that we dissolve the roots, the roots to get the copper using nitric acid. We tried also aqua regia, but apparently it's not there's no much difference between nitric acid and aqua regia. So we stick into nitric acid as the as to dissolve uh, the roots and get or extract the what we call this the copper in solution or put it in solution. So the setup would look like this. So we have an aluminum, what we call as the insoluble electrode. Then we have an acid solution which is laden with the copper. And we put in some variables like voltage and time. So you put in the current. So the variables would be voltage and time. So the results of the experiment, the copper in the solution was reduced drastically after the electrowinning process. This is noted at different time frames. Okay, we use 30 minutes, 60 minutes, and 90 minutes. And apparently, we also use the different types of currents. So we used 1.5 volts, 3, and 6 volts. Looking to the percent recovery, apparently there was no significant differences between the recoveries between 98.7% to 99%. So if we are to assess the efficiency, okay, considering the amount of energy used. So the most efficient condition are the variables 30 minutes and with respect to time and the current of 3 volts. So yun lang, 30 minutes and 3 volts. Kasi kung tatagalan mo pa, you consume most of the energy. So we try to use efficiency with low cost. So that's the result of the extraction of copper from the solution. So we were a bit successful on this. Now we try to recover arsenic. We tend not to use electrowinning for arsenic. We try to use another method, which is actually we call the hydrothermal mineralization technique. This is quite relatively new. It was used earlier in extracting metals from wastewater, but we try to use it in this process as well. So we use the accumulation, the arsenic accumulated in Pteris vittata and Pterogramma calomilanus. So with these two plants, we also dissolve them in acid, and we try to recover these metals from the solution. Okay, the concept behind this method is the natural hydrothermal process. So I think most of you have taken geology, maybe. You would know, you would come to this term hydrothermal mineralization process. Okay, to explain that concept, at depths of about kilometer or more, the Earth's surface, at a depth of a kilometer or more, magmatic and magma-heated waters circulate towards the surface. So these are very hot, acidic water. They percolate to the surface, and along the way, they dissolve metals from the different rocks. So these metals are now in the solution. Okay, they are dissolved in the solution. And if you change the condition of the system, for example, lowering the temperature or decreasing the pH, maybe, oh no, increasing the pH because it's highly acidic, yung magmatic waters, you increase the pH. Okay, maybe at that appropriate conditions, most of these metals would precipitate. So we're trying to use that concept So we try to mimic that concept. So in the process, we use stainless steel vessels, okay, to put in the solution. And apparently, well, the stainless steel are liner, may liner sila ng PIC, no? Yung poly, it's a plastic, a hardened plastic. Then we get to have changes in the variables like pH and temperature. From the equations shown in the slide, okay, to digest the arsenic of the plant, we used nitric acid. So it becomes in solution, and it is actually oxidized to arsenite. So arsenite, we need to convert this to arsenate, which is the preferred arsenic species for the recovery process. So arsenite to arsenate. The formation of arsenate should be consistent throughout the process. The hydrothermal solution, hydrothermal mineralization technique, actually we need to put in what we call a mineralizer. For example, calcium oxide, calcium hydroxide, to increase the pH. Kasi as we have noticed a while ago, yung solution is highly acidic. We try to increase the pH by putting in a mineralizer, which is, well, a calcium hydroxide. We also lowered the temperature at some point, and apparently we would expect, okay, same as a natural hydrothermal mineralization process, some of these metals to precipitate. So ginagaya lang natin yung natural process. So this is the setup. We have the stainless steel vessels. They are linered with peaks. Then we try to reduce, as I mentioned earlier, increase the pH and reduce the temperature. So at the end of the experiment, we were expecting some precipitates. So these are what we filtered, shown in the slide, some of the residue from the experiment. The results of the experiment was very encouraging. The arsenic concentration of the solution was reduced drastically, as you have seen here, with the final pH of the solution being more basic, well, which is actually we would expect as well. Apparently, the arsenic as noted to precipitate in solid form, which was actually we expect as well. This is an initial experiment, and we thought it would be wise as well to do further studies on this. Probably not only arsenic, but also different metals. Because in a natural system, it's not only arsenic that precipitates. Hello? Hello?
Yes, sir, we can hear you po.
Yeah. Since kailan yung last slide na you were not able to hear me?
Yung ano po, sir, yung start ng slide na to, sir. Nakamute po.
Saan?
Dito na po sa slide na to, sir.
Yung magnetic biochar?
Apo.
Okay. Previously, you can hear me.
No po.
What?
May-
I
maybe you can
think I
just-
had some technical problems now. Sorry for that.
Apo.
Can I, do I have time to go back to my previous slide?
Okay lang po, sir.
Can you help me out? What was the last slide that I missed? Na hindi nyo narinig.
Ito na po yun, sir. Yung
Ah?
copper recovery.
Copper recovery. So we'll start with that. I'm sorry for... Dito?
Sir, yung sa biochar po.
Yung
Yung
biochar
slide
po,
po
sir.
doon po nawala.
Oh, dito meron pa ako. You hear me? You can hear me?
Yes, sir.
So sa biochar nawala.
Yan na po,
Yes, sir.
So all the while, okay lang to? The
Yes,
previous
sir.
slides. Okay, very good. Okay. Let me continue. Sorry for that. Nawawala pa minsan. So you can hear me now?
Yes, sir.
Yes, sir.
Okay. So as I mentioned earlier, one of the experiments we were not able to do is this recovery of copper using magnetic biochar. I think you're familiar with this, but we haven't gotten into initial process on doing this. So actually, it is worth doing. And if you have some researchers with you, and I encourage you actually to explore on this method, magnetic biochar, to extract copper from the plants. Actually, I would like to collaborate with you guys if you have actually the time, or some of your students. I have all the plants you need. I have all the plants, and I can actually give them to you and for you to do the experiment. We do not have much of the equipment here at the Ateneo, but we try to collaborate with other universities to do this. Anyway, we'll talk about this maybe later. So after the recovery of copper arsenic, we try to apply what we already know for ecological restoration and mine rehabilitation. So the first thing we want to do is to propagate these hyperaccumulators. So kung wala ka namang a nursery, so we wanted to develop some nurseries here. So we constructed some nurseries to propagate these hyperaccumulators. And we try to produce as many of this so that we can have actually sufficient amount of plants that can be used for rehabilitation. We actually had a seed bank in Xavier University in Mindanao. And actually we use this to grow some of these ferns. And I think we were very successful as well in this seed bank. We have chosen a particular place in Manila Mining in Surigao, which is actually abandoned. And it is the Suyok copper gold open pit mine. So it is a relatively wide barren area. And the only plant that actually grows there is the fern Pteris, Pterogramma calomilanus. Very sporadic, very few. So if Pterogramma calomilanus can grow in that area, I think we can actually, well, we decided to plant, okay, physically plant Pterogramma calomilanus in that area as an experimental area. So we gather the Pterogramma calomilanus from the nursery and put them there. The challenge, okay, we encounter a lot of this. The challenge was on plant care management, as we found out that the fern is relatively sensitive to transplanting. Apparently, we thought it was a robust type of plant, but apparently it was so sensitive that we need to put in a bit of care, a lot of water and amendments and so on. So immediate care should be provided regularly, that of water and amendments. And if this is not done, a large percentage of the plant would eventually die. So hindi siya yung robust that we thought of. Well, we we did the experiment for a month in the area. So if you can see the slide, some have grown, but it was really unfortunate that it was not being monitored by our team regularly. So we left the experiment to the owner, the mine owner, to actually take care of the area. So we haven't gotten there, or we haven't been there for quite some time, but hopefully in the future we'll be there. They have informed us that some parts have grown, but a lot more has actually have died ng mga plants over there. But it was a challenge, but I think we need to be more careful about plant care management. So if we are very successful in this, what we call this, rehabilitation, and apparently we are successful also for the growth of these plants, we might actually venture into what we call phytomining. So phytomining is a concept wherein you plant or make a plantation of hyperaccumulators in an area. Okay, that is in addition to the purpose of decontaminating the soil, of which you can also use these plants, harvesting them and recovering the metals from these plants, which I have explained earlier. So that's phytomining. So both purposes of decontamination and recovery can be done. And it is possible. I just want to thank Ishard for funding this project. I know there are a lot more unknowns to be discovered, but it is a good start. I would like to thank the collaboration of LaSalle in one of the programs of one of the projects. Also, UP Los Baños for being there to help us out also in the program. So with the three universities, Ateneo and LaSalle and UP Los Baños, I think this project on rehabilitation using plants was a very successful one, although it needs to be put in more continuity to this. Okay, I think that's my last slide, and I hope you got to have some information, or you have gathered some information from that. And I would like to thank again for this opportunity to share what I know about phytomining and phytoremediation. Thank you very much.
Thank you very much, Dr. Claveria, for that enlightening presentation, and I'm sure the audience has learned a lot about the roles of plants in phytoremediation and phytomining, specifically the extraction of copper and arsenic. So now we will be having a 10-minute break until 10:08 a.m. If you have any questions, please drop them in the chat box with your name and your affiliation. So while waiting, let us watch a video about microalgae and their importance in the industry.
applications.
But found in a wide range of terrestrial and aquatic habitats, including lakes, rivers, oceans, and even wastewater, are the microalgae. These are prokaryotic or eukaryotic photosynthetic microorganisms that can thrive even in varying temperatures, salinities, and light intensities. With its photosynthetic capabilities, microalgae play an important role in producing approximately half of the atmospheric oxygen and some of the basic nutritional elements. Good day, everyone. I am Meredith Montalvo.
And I am Donna Belcena, and today's food for thought are the microalgae. Microalgae are found to be a rich source of several bioactive compounds, making it a suitable organism for different biotechnological applications.
But what's so special about this microorganism? Allow me to give you an example. Every day, the global population keeps on growing, thus the demand for fuel energy is constantly increasing. Although fossil fuels have been widely used in fuel production, they pose threats to the environment and are really unsustainable.
With this, scientists have looked for alternative ways to produce fuels in a more sustainable and efficient way. How? Through microalgae. As mentioned earlier, microalgae consists of different bioactive compounds such as carbohydrates, proteins, and lipids or natural oils.
Microalgae can be used in biofuel production through 4 steps: cultivation, harvesting, drying, and oil extraction. Cultivation can be done in a photobioreactor or an open system such as a pond. The algal cells are then harvested through centrifugation, filtration, or coagulation, depending on the type, density, or the size of algae that are being harvested. Drying is followed to efficiently extract the lipids through solar drying, convective drying, and spray drying. And lastly, the oil is extracted to separate the algal product from the dried biomass for the production of biofuel.
Aside from their importance in biofuel production, microalgae are also widely used in the bioremediation of different types of polluted water. This technology is called phycoremediation. This field emerged as early as in the 1960s when microalgae were used for tertiary treatment of municipal water. After the earlier works in phycoremediation, the field has evolved to many other applications in which microalgae and macroalgae are cultivated and utilized for specific bioremediation needs. More recently, a broad variety of non-pathogenic algae are being employed for wastewater treatment, including spirulina species, chlorella species, chlamydomonas species, oscillatoria species, nostoc species, and synedismus species.
Microalgae are really amazing. They can be used in treating polluted waters, while they also produce biomass that can be used for biofuel production. Therefore, the use of microalgae can be considered a green approach to environmental protection and sustainability.
Wait, there's more. Do you know that microalgae can also help us become healthier? Yes, it can. Microalgae are a rich source of carbohydrates, proteins, enzymes, and fiber. Besides, many vitamins and minerals like vitamin A, C, B1, B2, B6, niacin, iodine, potassium, iron, magnesium, and calcium are abundantly found in microalgae. Being such a rich source of essential nutrients, they are a major source of food, especially in Asian countries like China, Japan, and Korea. Green microalgae have been used as nutritional supplement or food source in Asiatic countries for hundreds of years. Nowadays, they are consumed throughout the world for their nutritional value. Some of the most biotechnologically relevant microalgae are the green algae, Chlorella vulgaris,
Haematococcus fluvialis, Dunaliella salina, and Cyanobacteria spirulina maxima, which are widely commercialized and used, mainly as nutritional supplements for humans and animal feed additives. Spirulina latensis, a blue-green algae, are gaining worldwide popularity as a food supplement, being one of the most nutritious food known to man. It is gaining worldwide popularity as a food supplement. It has been shown to be an excellent source of proteins, polyunsaturated fatty acids, pigments, vitamins, and phenolics.
Wow, with all that said, now I am really interested in microalgae. They are really useful. They can be a source of biofuel, they can be used for remediation of polluted water, and most importantly, they can be used as a source of vitamins and as food supplements. As a young person aspiring to become a woman of science someday, it really interests me to know that there are still a lot of things that can be studied and discovered in the field of biology.
Yep, that's right. There are lots of things to be explored yet in the field of phycology. I hope that you have learned a lot from our talk today and that you will be interested in performing research in some of the fields that we have mentioned.
All right, so now we will be proceeding with the open forum. Again, if you have any questions, please leave them in the chat box with your name and your affiliation. So to facilitate the discussion, may I call on Ma'am Erna L. Elias.
Okay, good morning, everyone. Thank you, Dr. Claveria, for your very interesting talk. So
You're
let's
welcome.
start the Q&A with this first question. It's from Danica Pamatian. If plant nursery was not successful in Suyok, what can you advise or what else you should do for the successful propagation of hyperaccumulators aside from looking into plant management?
Okay, thank you very much for the question. Actually, we were successful in propagating the plants. What we were not really successful, and we had some challenges, was the transplanting of these ferns into the areas we wanted to do some rehabilitation. That was the challenge, actually, and maybe the conditions are totally different. The area we wanted to rehabilitate is maybe totally barren, highly contaminated with metals, which is totally different from the nursery which we had where we propagate some of these plants. So we try to, what we call this, to use what we learned sa propagation and apply it sa rehabilitation. But again, it was quite difficult because the conditions are totally different. You put in a lot of water, maybe yung panahon na yon, hindi tag-ulan tag-init. Napakainit ng lugar na. Tapos you need to put in some amendments. So there's some sense of care that supposedly be inputted into the rehabilitation, which we try. Okay, I think this is one challenge. We try to look into parang another type of research we wanted to implement. It's not only extraction, not only propagation and assessment of copper and arsenic content, but also the rehabilitation process. It's not as simple as we thought of, but apparently it was a challenge. So I think I don't have any advice on, because I'm not a biologist. I have to admit that. Although they say, what some of my colleagues have mentioned is that there are plants that can be used initially in those specific areas, but they are not hyperaccumulators. Okay, they are not, but they can grow. We call them metallophytes. Then, if the conditions are now better after these initial plants or the primary plants, you can now plant your hyperaccumulator. Parang ganun ang dating. So it was really something that we can research on. We can do more work on it. So maybe if you are interested, you might be able to look into it as a research project.
Okay, thank you for that, sir. So I guess this next question can answer, can give a
Yeah.
little bit of contribution to that. So it's from Jericho, one of our RAs. So thank you so much, Dr. Rene, for the wonderful presentation. It is great to hear your group had done optimization of propagating these wonderful plants. Based on my experience in growing ferns, they need moist and humid environment to strive with little direct light exposure. So with this, are there any other bioremediating trees that can grow side by side with the ferns to provide cover and humid environment?
Yeah, that's perfect. Yeah, I think that's supposedly the approach. Yeah, if there are some, well, trees which are bigger, we call bigger, higher or bigger biomass that can actually grow, it will be helpful because it will help actually the ferns to grow. The moist content and the other conditions that require for these ferns to grow are indeed very important. So we have to be aware of those. In my experience, there are certain trees or plants with bigger biomass. They can actually survive in contaminated areas. Well, they are called metallophytes now. They are good as they're used for rehabilitation purposes as well. Pero ang gusto natin is that we wanted to propagate or to manage a number of hyperaccumulators. So we can actually clean more. We can achieve more of what we call this decontamination, and also extract these metals from the plant. So that will be a good thing to work on. Maybe plant side by side certain metallophytes that can tolerate the contaminated area, and they grow very well. And comes with it, accompanying it will be these hyperaccumulators, which actually extract the metals from the soil. So we can have both actually. So that's a very good thing we might want to look into. Thank you very much for this observation.
Okay. Thank you very much, sir. So we have a few questions. Here in cell biology, it is known that subculturing causes alterations in genetic expressions. Might this be applicable to plants? Are the propagated ferns already tested to be effective in phytomining? Are they as effective hyperaccumulators as the ones that grew naturally in the mining site? So three-part
Yeah, very
question.
good. To be honest, no, the propagation experiment was actually done. I would like to acknowledge one of our team from Xavier University who did the propagation. Actually, this was published in journals, the propagation of Pteris vittata, and it was well accepted. well, I cannot give you much of the process on how they did it. But maybe I would agree with you that maybe there's a difference sorry, between these cultured plants against the natural plants that grow sorry, in this contaminated soil. Maybe there's some difference there. So it's worth also looking into that. Yeah. What could be needed for these propagated plants to survive in rehabilitation areas? That's a very good point. I think we have to consider.
Okay, thank you, sir.
Yeah.
Another one is, for how long can the plants absorb metals while staying free of signs of toxicity?
Actually, these ferns, if they are in their natural habitat, they can grow to maturity. I don't know how many years. Yun nga, the process is that there's a question raised by a colleague of mine, also a geologist, mentioning that if these plants grow into maturity, they die out eventually, right? And the arsenic or copper absorbed in the plant goes back into the system, to the soil, and so on. So it's a cycle. The metal is not removed, but actually circulating. So I think as long as the plant grows or thrives very well, there are a lot of metals being accumulated. So we thought, if they are able to accumulate so much of these metals, why not harvesting them? Okay, what I mean harvesting is that you just remove the above ground. So that's why we call it hyperaccumulators. You cut the above ground component. That's where you harvest or recover the nickel. So more likely you leave the roots and some of the stem in it, and the hope that it will grow again. Parang ganun eh. Parang you harvest, don't remove the roots, take only the above ground components, remove yun yung concept namin for the experiment. Remove the metal from the above ground components, and you get the metal. Then you can dispose the waste eventually. It's not like the normal cycle that if the plant dies, the metal continues to be circulated into the system. So we try to remove that one, the one component of the system, and we benefit from it because we get the metal from the plant. So I think that's the concept we wanted to use, and we call it plantation. We put it in a contaminated area. We make a plantation on that, and we harvest the above ground components. Let the plant grow again. Parang ganun. We were hopeful to do that, but of course, there are some constraints. But if somebody can do it, why not?
Yeah. Yes, that's a very interesting concept.
Yes, very interesting.
Parang farming na.
Yeah, farming.
Mining at the
that's
same
very
time.
good. Yeah, farming. Yes, yeah, very good. Very good.
So another question is, since there are already species that are proven to be hyperaccumulators, has there been any proposal or government policy made to use phytoremediation widely in rehabilitation of mining sites? Would you know?
I think the output of the project we had with Pishard was a successful one, and apparently we just need to implement what was already done. For example, I'll give you a background on the other universities, what they have done. There are three components kasi yan. The LaSalle component they were looking into what we call these bioindicators. So there are plants, metallophytes or mainly metallophytes, that will inform you that there is so much metals in the soil. So based on their anatomical components or their features, it will tell you that ito masyadong mataas yung metal content niya sa leaves because of these features and so on. So they did a lot of work on that. So if you encounter a plant with certain features, you might say that, okay, this is very high in nickel, this is very high in copper or arsenic. So that's the LaSalle component. For the UP Los Baños component, they focused on nickeliferous laterite mining, wherein I think they were able to discover, if I'm not mistaken, 4 new hyperaccumulators published. They published that. So actually, we are now known, the Philippines is known for a lot of nickel hyperaccumulators, not discovered yet. So we're discovering a lot of that. What they did also is to propagate these hyperaccumulators in lateritic soil. So I think they were a bit successful on that because ang plants ng nickel hyperaccumulators are of bigger mass. Yung biomass niya malalaki. So they're partly trees at some point. So I think they did that. Yun sa Ateneo naman, we focus on extraction process. So we tried nickel for the initial process, but the only process we thought of in extracting nickel from the plant is incineration. And bawal naman ang incineration sa atin. So we thought it's better to use wet chemicals na lang. So we focus on copper and arsenic. So yun, it came about that based on the 3 programs or the 3 projects, I think we can implement much of this mine rehabilitation throughout the Philippines. There are a lot of what we call legacy mines, okay, here in the Philippines. Legacy mines meaning these are abandoned mines, no rehabilitation at all. The owners left and the soil is highly contaminated. Walang tumutubo dyan except for certain indigenous plants that can actually survive. So one of these indigenous plants are the ferns. So we thought ferns would be a very good, what we call these plants that can be used for rehabilitation, particularly on copper and gold mines. So I think if we have the funds or the mining companies or industries can help out in doing rehabilitation, because actually they are required. They're required to rehabilitate their own areas. So we are actually providing them some options to rehabilitate. So I think if this is acceptable and you get into the process with enough support, I think it will be successful throughout the Philippines, this mine rehabilitation. And I think it is one of my advocacy kasi as a geologist, it's not that I look only for new mine areas, exploration, but I also wanted to do rehab, particularly those that are already abandoned. This is bad, what do you call this? Abandoned mines are not good to the eyes dito sa atin. So I think we need to do that for the environment.
Okay, thank you, sir. So what is your group's plan for wide scale application? Do you have a plan for wide scale application?
Well, sorry. After the program, actually yan ang problema sa mga projects. Kapag tapos na, wala nang follow-ups. So I think if only we can ask further support from the government and with the support also from the mining industry, maybe maybe they can we can have this as a long-term process. Ang problema lang kasi for now is that we go into the short term lang. There are some mines which actually rehabilitate their areas using exotic plants. Exotic plants meaning they are not indigenous in the area. So exotic plants are they are a bit robust and they grow anywhere, but they are not meant to be there. So as I agree with a lot of biologists, is that do not plant exotic plants in areas, plant what is indigenous, because the ecological system will be compromised in case of you put in a lot of these exotic plants. These are very short-term methods, and what you were thinking are more of the long-term. So support is needed.
Thank you for that, sir.
Yeah.
We still have many questions on ganda ng discussion. However, we're out of time. So can we just send the questions to you,
Yes,
and
please.
then
Yeah.
you can email them directly, or you can send the replies to us, and then we can reply to them. So we'll
Yeah, sure.
just collect the questions, and if any of our participants can think of additional questions, just send them to us, and then we'll pass it on to Dr. Claveria. Is that all right,
Yeah.
sir?
Thank you very much. Yes, please.
Okay, so thank you very much, sir, for answering our questions. Ang ganda talaga ng discussion. Sayang po, kulang tayo sa oras. And thank you everyone who submitted questions. So Meredith, back to you.
Thank you so much, Ma'am Erna, and to everyone for all the questions raised. So before we award the certificate to our speaker, if you like webinars and discussions such as this one, don't forget to like and follow the socials of the BRSL and the NSRI. You can find us on Facebook and YouTube, where we share updates in our services, as well as news and innovations in any field of biological science. So again, our biggest thanks to our speaker, Dr. Claveria, and to show our appreciation, may I call on Ms. Zarina Katapat to present the certificate.
Hello po. May I ask Dr. Pai to please open your camera to award the certificate po? Ma'am Pai. So the Biological Research and Services Laboratory awards the Certificate of Appreciation to Dr. Rene Juna R. Claveria for having delivered a lecture on the BRSL Webinar Series number 9, Bio at Work, the Roles of Plants and Microbes in Environmental Remediation, held on September 34, 2021 via Zoom, signed by Dr. Pierre Angeli G. Vital, Head of BRSL, and Dr. Elmer S. Estacio, Director of NSRI. Again, thank you very much po, Dr. Claveria, for joining us in the 9th installment of the BRSL webinar series. Thank you po.
Thank you very much. Thank you very much for this.
We are now nearing the end of this webinar, and to give the closing remarks, may I call on the head of the Biological Research and Services Laboratory, Dr. Pierangeli G. Vital.
Thanks, Mary Dave. Good morning, everyone. Good noon, good afternoon, or good evening for some of you who are attending in other parts of the world. So I'm sure we are all having a great morning or afternoon or night. So just please allow me to thank the following for making us reach episode 9 today. So first, of course, our speaker, Dr. Rene Claveria of Ateneo de Manila University, for graciously accepting our invitation and sharing his expertise with us regarding the role of plants in bioremediation. I'm sure you learned a lot, and I definitely learned so much. The different phyto words, lahat ng phyto po na narinig ko kanina. And noted Sir Claveria on the intent of collaborations. Hopefully in one way or another, we can collaborate, or even someone from our institute po. So thank you very much po again, Dr. Claveria. So second, of course, this webinar will not be possible without the support of our NSRI Director, Dr. Elmer Estacio. You saw Dr. Elmer po kanina. So thank you very much, Sir Tux. And this webinar is special as it is the culminating activity of our interns. And nakikita nyo na po yung interns natin mula kanina who are MCs from University of Santo Tomas. And in relation to this, let me thank also the Office of the UPD College of Science and the Office of the Associate Dean for Research, Innovation, Development, and Enterprise. I hope I got that correctly, or OAD RIDE, for making the BRSL internship possible. Third, of course, this webinar, as well as the preceding 8 webinars during this pandemic, is due to the efforts of the remarkable researchers and staff of BRSL. You can see their names po and pictures shown on the slide in your screen right now. Marami pa po kami sa lab, no? But these are the people, the hardworking people behind the episode 9. So again, if it's not too much to ask to everyone, please click the memoji or the reaction heart button to show your appreciation or clap po to everyone. Thank you. Fourth, and most importantly, I would like to acknowledge our hardworking interns. As I have mentioned earlier, sila po yung mga behind the scenes, and nakita po natin yung MC kanina, albeit virtual. The students from UST, Kiana Dominic Carrion, if you can turn on your cam so you can be seen by everyone. Maria Ara Janica Ferrer, Janiel Mesde, and Meredith Eunice Montalvo for helping organize the BRSL webinar episode 9. Again po, please round of applause for these guys. Kiana, Ara, Janiel, Meredith, I hope that you continue to be involved and pursue a career in the field of biology, as there are a growing number of challenges that science, particularly biology, can address, such as, of course, our theme in this webinar, remediation. I can't wait to see you successful in this field. So earlier, you already saw the available resources and services that our lab, Biological Research and Services Laboratory in the Natural Sciences Research Institute, University of the Philippines, Diliman offer. Although our lab is still physically closed, na-mention na po ito kanina ni Dr. Estacio, we are still accepting online consultations and continue to organize webinars such as this, as well as, of course, online workshops. So please feel free to email or contact us if you have biological concerns. And please stay tuned with our BRSL Facebook page for updates. You can see our contact information flashed in your screen right now. Lastly, of course, I would like to thank all of you, our participants coming from all parts of the country and the world, whatever your time zone is, either waking up very early or staying late at night to join us, who show great interest and enthusiasm in biology and its allied fields, and joined us this morning, again, maybe afternoon or night for you, for the BRSL webinar episode 9, Bio at Work, the role of plants and microbes in environmental remediation. With this, I wish you to have a wonderful and safe day and weekend ahead. Hope to see all of you again in our future activities, such as Episode 10 next month. This is sort of an advanced announcement. We will be having, again, another interesting topic. So thank you very much. And I turn back over the virtual floor to Meredith.
Thank you so much, Ma'am Pai. So this webinar would not be possible without the team behind it. Again, job well done to the BRSL interns and the research associates who tirelessly planned this webinar. And of course, to you, our audience, all thanks for taking the time to join us today. If we may invite everyone to open their cameras for the photo documentation.